Alright, SBI3U. This is video number 4. This is about genomics and biotechnology. So there it is back there. That's the note that we're working on. Here's our learning goals. First off, what the heck's a genome? Second off, we're going to talk about restriction enzymes and gene editing. You might have heard of CRISPR. We're going to talk about CRISPR. Then we're going to talk about fingerprinting, like DNA evidence. And then finally, being able to trace like evolution through the genetic diversity, through the genetic changes that have happened in a population. So get that out of there, and we're just going to jump right in. So genomics and biotechnology. First off, what the heck is a genome? So, here's the keep in mind. The genome is all the DNA. It's not just the genes, which is kind of weird to think about, but yeah, it's all of the DNA. Not all the DNA is actually in genes, and a genome actually comes from two words that a botanist back in the day just kind of smushed together. Genome he took the word gene and he took the word chromosome and he just kind of just mushed them. So up there you'll see gene plus chromosome, that's genome. Gene comes from the Greek genesis, right, for to make. And the some part of chromosome actually comes from the word for body, like somatic cells and stuff. That's the word for body. So genome, if we kind of parse it right, could be like the body of genes, or maybe the things that generate the body. The genome generates the body, maybe something like that. So, you can kind of think of it that way. But a genome is all the DNA, all of it. Sequencing is how you figure it out. So, like, the sequence is like A, C, T, C, G, D, T. Everyone's got a specific sequence of that stuff. And you can sequence a gene, so just the gene. You can sequence a chromosome, or you can sequence the whole genome. And that's what they tried to do. They tried to sequence one particular human's genome, and they launched that in 1990. They wanted to sequence the whole genome, but at the same time, they also wanted to work on improving the technology for sequencing. They wanted to sequence genes that lead to certain diseases. They sequenced bacteria and viruses and stuff at the same time. So it's not like they only worked on the human genome. They actually got a lot of extra tech out of it. But nonetheless, it took them 10 years. They did accelerate genetics by a lot by doing this. They also spent like $3 billion pulling it off. Now, after another 20 years of advancement in this technology, you can actually now get your complete genome sequenced in 2 weeks for about $1,000. You can get all 3 billion base pairs of your specific genome. You can get your code. You can get the source code. So what they do is they get you to just kind of take a cheek swab, send it back to them, and then they do the work and they send you back your genome. So if you got $1,000 lying around, maybe you want your entire genetic code. I don't know. If you don't have $1,000 lying around, here's the actual website here. If you don't have $1,000 lying around, you can spend €300, and that'll be 8 weeks. So it'll take 8 weeks to get your genome. Yeah, you have to wait a whole 2 months to get all 3 billion bases, and you get like a whole range of things that they'll check for, all these different diseases and stuff. They'll tell you, how well you'll react to certain drugs, and what kind of risk factors you have for different things, and different sports injury risk for some reason is in here. Like I guess they claim that they can tell you how best to work out based on your genetics and stuff like that. I don't think the art of genomics has gotten that far yet, but maybe it'll get there soon. We've even got this Gene Plaza, geneplaza.com, which, if it loads, here we go. It's a marketplace for genetic reports. You upload the genetic report, the sequence that they gave you, and then different people can actually can use it and tell you different things, like math ability, based on genetics. Yeah, it's weird, man. This one's my favorite. 122 Shades of Grey. The R used to not be in this word. So, yeah. This app, disclaimer, this app does not predict same-sex attraction. Let's just zoom in on that for a sec. Disclaimer, it does not. The sum of the effects was something like 3% of same-sex attraction. So, yeah, all this does is tell you if you have the genes that might determine 3% of your sexual orientation. But hey, you can buy it, I guess, if you have genetic data to sell on Gene Plaza. Yeah, this is where we're at, people, where we've got apps for genetic information now. Yeah, always freaks me out. Anyway, yeah, if you got $1,000 lying around, maybe get your gene sequence. Maybe don't. Maybe spend it on something else. So yeah, we got 3 billion base pairs. That's why it took so long, because 3 billion is a lot of things to sequence. We got about 20,000 genes, so every protein in your body needs a gene. So even down to each individual smell receptor. So you have like a smell receptor for raspberry, that's one gene. A smell receptor for strawberry, that's a gene. A smell receptor for, I don't know, roses, that's a gene. Things like that. Yeah, so that's made up of coding DNA and non-coding DNA. And here you see this is the same figure from last lesson. Only about 1.5%, less than 1.5% of all of the genome. This is all 3 billion. Less than 1.5% actually codes for protein. Everything else just kind of codes for random stuff, but it's still part of the genome, and it still had to be sequenced. This red part tends to be really well conserved, because if you screw up a protein, it's usually bad for you. But these ones will get mutations pretty often, and so when we talk about DNA fingerprinting in a second, realize that mostly it's in this random DNA that can mutate without really any trouble. And that's why people have such different bands of it. That's why people have such different DNA between them. We're actually analyzing the 98.5% of the genome that doesn't actually code for anything. Yeah, here's some other genomes. If you thought humans were really complex and had a lot of genes and that just makes us really cool, well, got news for you. Wheat has 17 billion base pairs. So we have 3 billion base pairs, like A, T, C, 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 whatever. Wheat has 17 billion. So that means that in terms of the amount of DNA, wheat has almost 6 times our DNA. So, maybe 6 times more genes? We don't actually know. Yeah. I don't think we've done a wheat genome yet. Someone probably has. It's so easy now. A salamander has 84 billion, so even crazier. If you want to compare yourself to an animal, a salamander has, more than 20 times more DNA than you. And the amoeba is way crazy for a size of their genome. Every time they replicate, they have to replicate 600 billion base pairs. And we think, okay, well, maybe it's the number of genes that matters. Well, rice has 50,000 genes. We only have 20,000, right? Mice have more genes than us, stuff like that. And we're talking specifically about that slice, right? Maybe that slice is bigger in a mouse than it is for humans. Yeah, so that's a genome. That finishes our first section, right? We're going to then talk about biotechnology. Right, we've done what's a genome. Next up, restriction enzymes and gene editing. I'm going to pause while I fix up the keep in mind and the topic. And I'm back. So I've fixed it up. Now we're talking about restriction enzymes and CRISPR down below, and talking about the different kinds of restriction enzymes and how we're going to use them. So yeah, let's jump in. Here's what a restriction enzyme does. And I should move my thing over. So here's what a restriction enzyme does. It's a little enzyme, and it chops DNA at a palindromic repeat. So a palindrome is something that's the same forwards and backwards. You can think of like taco cat, which is taco cat forwards and taco cat backwards, or race car is another famous one. So here you see this is GAATC, but then backwards it's also GAATTC, so GAATTC forwards, GAATTC backwards. The restriction enzyme, the specific restriction enzyme, because there's different ones that might recognize that, would go in and chop that and separate it, and it would leave almost like a sticky end, like a little loop. And then if you had another piece of DNA, maybe from a different organism, that you cut out with the same restriction enzyme, then those two could actually join together, and you get a new DNA. Like, you know, you're just kind of gluing it like that, right? So you cut one strand of DNA to make the sticky end. You use the same enzyme on a different strand of DNA. You join them together, and then because DNA is DNA, you don't know that one came from one came from the other, or the organism doesn't know that the foreign DNA came from a different organism. So how was this used? Well, in 1978, so that's a while ago now, 1978, right? 42 years ago, they did this to E. coli. They took the insulin gene out of humans and they chopped it with a restriction enzyme. See that like sticky end? That sticky end? And then they took E. coli, and they have this round piece of DNA, and they chopped that plasmid with the same restriction enzyme, and it opened up. So, like, you know, they took the human DNA and they took the plasmid and they opened it up, and then they stuck the human DNA into the plasmid. And now the E. coli doesn't know any different. It still looks like any old DNA, but now it's got the human insulin gene in it. So it's going to make insulin. And then you can, you know, let the E. coli grow and grow and grow and grow and grow, make lots of, you know, E. coli babies, and then, you know, extract the insulin that they're all producing, because E. coli doesn't have any use for insulin. It's a human hormone, but humans sure have use for insulin. So you can make human insulin from bacteria. Very good use of restriction enzymes. And again, you can see that sticky end that we were talking about, this sticky end there, that's what that's trying to show. So they did that 42 years ago, and we've been using bacterial made insulin ever since. So, you know, if anyone ever starts talking about GMOs, just ask them if they know any diabetics, and then ask the diabetics about the GMOs. This is a genetically modified organism, and in fact a transgenic organism. That's kind of cool. Now this one's got a bunch of good press, well, maybe good press these days. This is CRISPR-Cas9. You might have heard of CRISPR. It's like the holy grail of genetic engineering. The reason that CRISPR is so cool is the restriction enzymes. Restriction enzymes need a specific palindrome to work on. Like this one is GAATTC. CRISPR is this protein that comes attached to this Cas9 thing, and the Cas9 can have this strand of RNA that can be whatever sequence you want. So it will specifically match to whatever sequence you want it to find, and will cut that very specific sequence. It doesn't have to be a palindrome. It doesn't have to read the same forwards and backwards. You're not stuck with just taco cat or race car. You can literally put anything and find anything and chop anything out. And if you chop anything out, you can also put anything in. So that's the power of CRISPR. It's like a laser guided pair of scissors. It just chops out exactly the gene that you want and can insert whatever gene you want. So that's really powerful. And it opens the door to, for example, gene editing a living organism. So here's a very recent news story actually. The OHSU, it's a site unit in the States, on March 4th. March 4th, so it is March 24th. This was 20 days ago at the time of this video. They used CRISPR in a human body. So this was an adult human who had this, he has this genetic disorder that leads to progressive blindness because he has a bad gene for one of the eye receptors somewhere in there. And what they did was they decided to use CRISPR to fix it. So this like blue thing, the Cas9 nuclease, they would have grabbed that sequence for that specific gene for blindness and added it to the CRISPR molecule. And then the CRISPR molecule would have gone directly to that gene, chopped it, put in the right gene with a different technique, and then now hopefully, if they do that just in the eye cells, his eye cells will now have different DNA than the rest of his body, and hopefully it'll be the right DNA. You can actually see them injecting it, up here. See them trying to get to the optic nerve and then trying to get it to go. But yeah, that was 20 days ago. We had our first in vivo CRISPR gene edit. And I'm sure there'll be more to come if this works. They said that it would take about a month to figure out if it worked or not. So maybe, if you're watching this later, maybe check to see if they got it right. I don't know. I have no idea right now. I checked the newsroom that they have on their website, and I don't know yet. Maybe you do. Such is the future. All right, next up, we have done genome. We've done gene editing. We specifically talked about E. coli and the CRISPR for humans. Now we're going to talk about DNA fingerprinting, which is done mostly for crime scenes and for paternity tests. Again, I'm going to pause and change the things here and here. All right, so we've changed those. Our current topic is now DNA fingerprinting at crime scenes. DNA fingerprinting at crime scenes. And our current keep in mind is DNA is evidence, but it is not proof. There's always a chance that two people have similar enough DNA that you get confused for someone else. There's always a chance that, your DNA is there, but it's not because you murdered the person. Maybe you just, were over there playing video games last night and one of your hairs is on the couch or whatever. It's evidence that someone might have been there, but it's definitely not proof. So, it's very good evidence, but not always proof. So this is a biotechnology. It uses this thing called gel electrophoresis, and it creates this pattern of bands on a gel. There's like a fingerprint that's unique for each person. And the DNA that they're specifically looking for is the number of repeats of these random repeat sequences. So I'm going to jump back through the slides a little bit to this slide back here, right here. See these like repeats. Those random repeats are what they're actually looking for, like how many repeats there are and stuff like that. That part of the genome, because it doesn't code for any protein, if there's a mutation there, it doesn't really affect you. So people will accumulate those mutations pretty quickly, and so it leads to a relatively easy way to determine their differences. This is what these bands look like, and let's talk about how it works. First, you got to get DNA, a sample of DNA. Then you grab restriction enzymes, so those same palindromic ones, right? Maybe it like GTTAAG or whatever, and it chops it into fragments, and the fragments are going to have different sizes depending on the person's DNA. It's however long it is until the next GAACCG or whatever that I said earlier. So, one person might have a short distance between two of those, one person might have a long distance between two of those, and so they'll have different sizes of DNA. Then that goes into a gel, and they put electricity on each side of the gel. They put a positive on one side and a negative on the other side. And then the DNA, because it's charged, will move. So DNA is negatively charged. I mentioned that in the last video, in video 3.3, that the DNA, the phosphate groups on DNA have negative charges. So they're going to run away from the negative charge and towards the positive charge. But they're trying to push through a gel, right? They're trying to push through a a kind of a dense material. And so the thinner ones, the smaller pieces, are going to be able to push through faster than the bigger ones. They won't be able to push through as quickly. So the smaller ones are going to actually go further, and the bigger ones are going to go less far. So here's the diagram, right? There's going to be a negative at the top and a positive at the bottom. So DNA being negatively charged is going to run away from the negative, because opposites attract. Positive attracts the negative. The same charges will repel, so they're going to run away. Larger fragments get left behind because they're just big. They can't push through the gel as quickly. Smaller fragments, being smaller, will go further. Yeah. The gels are pretty fragile. There's some more kind of stuff here. Right. The gels are fragile, and they need to be supported. They also don't last very long because this gel, I've made these before. They kind of feel like really thick Jello. If you've ever made Jello, it's a very similar kind of feel. What they do then is you have to put some kind of marker on it that that lets you see the DNA. When I did it in my in the lab that I was working in, we would use a fluorescent marker, like a chemical that would glow under ultraviolet light, and so we didn't need to do the X-ray thing that's mentioned here. We had an ultraviolet one, and so I would just make the gel and then go to a different room, and there was this really strong ultraviolet light, and it would shine. Like a glow-in-the-dark thing, and the DNA bands would pop up, and I would take a picture of them. And it was really interesting. Right, so you see this is the whole summary of it. You cut it with the restriction enzymes. Different people get different sizes, and then they move down the gel. Bigger chunks get left behind. Smaller chunks go further, and then different people have different bands. So if you had like a suspect and then the criminal's DNA at a crime scene, this wouldn't be the person because the restriction enzymes have cut up this person's DNA differently than this person's DNA. So a different kind of DNA. Right, so another picture of that summary. The same idea, you can see the gel, the way it works there. All right, this has applications in forensic science. It's been a little over 30 years, maybe 33 years, that we've been using DNA as evidence in court cases. The first person to ever be declared innocent because of DNA was this guy Richard Buckland, and this other guy was can't say proven, but because again DNA is evidence, it's not proof. But along with other means, motive, opportunity stuff, this guy Colin Pitchfork was proven to be the real killer. So that's the thing. DNA is evidence; it is not proof. With that in mind, let's play whodunit. So we've got the victim's DNA that we did the gel electrophoresis to. We did the suspect's DNA, suspect 1, suspect 2, and then we've got the stuff that was left at the crime scene. So who did it? And I think it's pretty obvious. Suspect 2 was clearly at the crime scene at crime scene number 1. Suspect 2 was at crime scene number 1. But it doesn't look like suspect 2 was at crime scene number 2. What we see at crime scene number 2 is the victim's DNA. The victim's DNA is at crime scene number 2. So this suggests that suspect 2 was there. It doesn't mean that suspect 2 did it, it just means suspect 2 was there. Suspect 1 wasn't at either crime scene, so we can just let suspect 1 go. Suspect 2, we might want to question a little bit about, hey, we have proof that you were there, kind of thing. And here's another one. This one's a little bit more fuzzy and a little bit, more interesting. Here's the crime scene. Which one matches? Well, I think it's pretty clear. It's suspect number 2. So the advantage of DNA fingerprinting is you get these very clear images. All right. Here's another one. This really looks like a real gel. This is what a real gel kind of looks like. Here's the victim. Here's the specimen that was taken. Maybe it was like some I don't know. Maybe the victim like scratched and they found skin under the fingernails or something like that. Which suspect matches? Well, obviously it's suspect 1. But you can see here though that that these bands are like obviously in the same place with the same pattern, but they don't look exactly the same because each gel runs a little bit differently. So so again, you can see the very clear matching, but they're not exactly not exactly the same. Yeah, and once again, not necessarily that the suspect did it, just that matches the specimen that was found. All right, now the other thing is paternity. Paternity is slightly more complicated because what we're doing is looking for who's the dad in a kid that has half dad and half mom. So DNA fingerprinting for paternity. And still the same, keep in mind up here that DNA is evidence, is not proof. So usually linking parents to children, you can figure out who's who. Normally it's used for paternity because the mom is normally known, but it can be used for maternity as well. It's not necessarily for paternity. For example, if like babies were switched in the hospital, you can use genetics to figure out who's the real biological parents of the baby. So here, unlike the crime scene thing, we're going to see part of both. So here's Mary, who's the mom. Here's the kid. Is Bob or Larry the dad? Right, and it's Larry. Right. Bob and Larry both have this size of repeat, but only Larry has this one and this one. And then this one came from mom. This one clearly came from mom. So that's the one. Here's another example of this. Right here, is it male 1 or male 2? That's obviously male 1. The thing that makes it different from fingerprinting at crime scenes is that the kid is going to be a mix of both mom and dad. So the repeats, you might actually see missing repeats. Like here, this one is missing because maybe there was a different section that was like shorter and it went further, or something like that, because the kid's DNA is a mix of mom and dad's. Or maybe that's on the chromosome, the chromatid from dad that didn't end up in the kid. Remember, only half of dad's DNA goes in. So maybe this was from the other half. Same thing with this one for mom, right? That didn't end up in the kid, that didn't end up in the kid, that didn't end up in the kid, and neither of these ended up in the kid. But that's that independent assortment thing during gametogenesis. All right, here's one more. These look more like real gels. Here's the mom, the child, who's most likely to be the dad. And it's probably no surprise, it's male number 1, because you see this band, that band, that band, that band, that band. It's a lot more likely to be male number 1 than it is to be male number 2. Again, you can't be absolutely sure, but it is male number 1. And you see the mom is here highlighted in purple. All right, now the last piece. This is the last part of this lesson, and that's DNA as evolutionary evidence. All right, DNA as evolutionary evidence. And hey, we get to keep the same keep in mind up there. That DNA is evidence, but it's not proof. So what these are, they're called holotypes, 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 and they are pieces of DNA that tend to stick together when parents have kids, like genes that are associated closely with one another and that take a long time to change, like millions of years to change. And this is a map of human migration patterns based on these common holotypes. You see like M3, M130, M242, all of those are like specific clusters of genes that like to stay together. And changes in them indicate branches. So like M45 branched into M242 and M173 and stuff like that. And if you trace it, you can actually start tracing backwards, and we end up with, Africa, right? The cradle of life. And that's where humans are thought to have originated. That's where the oldest kind of human remains are found and stuff like that. And you get a map of where humans went over time, where different populations. So one population speared up north towards Europe, another one went along like to India and Indonesia and China and stuff like that. Somewhere along the way, part of them split off and went north across Siberia, and then those are the ones that ended up in North America. There's a whole subset that just went off into the ocean. And you can kind of trace it evolutionarily in terms of a DNA history of humans. And when you combine that, remember that that DNA is evidence, but it's not proof. But when you combine it with archaeological finds from human remains in these areas, you can build up a pretty good history of human migration. Some of the neat things here is you can see, like there was a group that went kind of to India, and then kind of hopped across. Instead of coming up through the mountains here, they actually just hopped across the plateau. Or that same group actually ended up going from South Korea to Japan and then up to China. Stuff like that. It's neat. You can learn some interesting things just tracking the DNA, and then you can start looking for evidence that supports that theory in archaeological digs, in expeditions from there. That's it. That's everything I wanted to talk about today, right? We want to talk about what's a genome. We want to talk about restriction enzymes and gene editing. Talk about DNA fingerprinting. So crime scenes and paternity, they're slightly different. Crime scenes, you want to match the DNA exactly. Paternity, it's both mom and dad factor into the baby, so it's like a partial match on both. And then tracing evolutionary history through genetic groups. I'm going to put links to the websites that I showed you down in the description, so if you want to check those out, please do. And please don't get caught up in the hype for Gene Plaza. Like I said with the 122 Shades of Grey app, it's like they can maybe match 3 or 4% of any particular ability. So the Are You Good at Math Gene app is probably not biologically significant. At least not yet. Who knows what the future holds. That's video number 4. Thanks for listening, and I'll see you guys in class.